Hello everyone. This is my presentation topic for the food science project. The research article, the capability of tyramine production and correlation between phenotypic and genetic characteristic of Enterococcus faecium and Enterococcus faecial strains. My name is Abrar Vohara, and I am going forward to provide some information about these research topics. So let's move on. So first of all, we need to understand what is the tyramine production. Okay, so as the tyramine occurs widely in plants and animals, that is metabolized by the various enzymes. So it includes different things. So it includes the monoamine oxidases and the food which contain means like a more amount of salt and acid, which we are usually the pickle, curd, and aged have the more amount of tyramine production because it's mainly correlate with the pH of the food product. And okay, talking about the tyramine in the food, and it is often produced by the decarboxylation of the tyrosine during the fermentation or decay. So we can say that tyramine production is mainly correlated with amounts of acid produced by the food, like in generally the lactic acid is mainly correlated with the tyramine production. So it is important to know that what is the tyramine production in this research article, because this is the base of this research article. So now moving forward to the main part. So in the different food, there are the different ways by which the tyramine can produce in it. As I said earlier, the lactic acid bacteria is the most efficient producer of tyrosine decarboxylase, the main what you call the enzyme which are responsible for the tyrosine formation in the food system. Apart from that, the lactic acid bacteria belonging to the genus Enterococcus are recognized as the most efficient tyramine producers, in the food. If these acids are present, so while they are means curdling and all that, while like a lactic acid bacteria from the different food, when they are in the stage of like a pickling and curding and aging, so they are producing the a different type of the acid by which the tyrosine can produce inside the food, and is also accumulated by the enterococci in higher amount that is already occurs in late exponential growth before the stationary phase. As the stationary phase means moving forward for the exponential phase, the tyramine from inside the microbial cells through the action of TDC. Okay. Uh, this is the main abstract. The main aim of the study of this entire experiment was to find out the diversity of the tyramine production and its capability of, about the 4 different Enterococcus strains in relation with the genetic characteristic and different environmental condition, by which we can know that the tyramine production can means mainly correlated with its genetic modification or like a genetic characteristic of the different foods, and in which we take the 4 different strains of the Enterococcus as per the methodology experiment is carried out, and based upon that the genetic organization and nucleotide sequence of these operand were highly conserved in Enterococci strains. So depending upon the different strains and different experimental condition like a pH and incubation temperature and all that, we can observe the result after that. And on the opposite side, a codon stop was identified in the translated tyrosine sequence of Enterococcus faecium. So in the present research contributes to find out the genetic characteristic of enterococcal strains, and it's mainly correlated with the specific mutation to their different strain developed by the tyraminogenic activity. So we can say that after taking different 4 strains and means observing and performing these 4 strains depending upon the different environmental condition like a temperature, pH, and different incubation temperature, so different results were obtained. And after that, in the different result you can find out that these are the strains who are the most means capable of producing the tyramine production, and these are the means Enterococcus strains which are not producing so much amount of tyramine production. So we can know that these can be correlated with the different environmental and genetic characteristic of the food. The materials and method we used to carry out these experiments, like experiment was four different strains of Enterococcus faecalis. Different name is there in technical, and they were suspended in the different buffers which was already prepared. The buffer used were the citrate buffers having the different pH and the sodium fluoride concentration, and during all the experiments we used the different incubation temperature for each of the four strains, and time to time the Tyramine production rate and cell viability were observed by the plate counting and HPCL like analysis to know that how much means Tyramine production occurs in each strains and how the cells is means generated or means obtained in a different time period. And we have to observe the activity of the purified tyrosine decarboxylase, which are the main means producers of the tyramine. So we can know that how much tyramine means produced by each of the strands, and after all different methodology and experiment, results were taken into the consideration, which reduced the loss in cell viability was observed in all tested condition except the like a pH after the for 24 hours. So different methods about the four different strands, and results were obtained, and apart from that, and different means condition were used to find out means the rate of the tyramine production, and different NaCl concentration were used to know that how the tyrosine is produced and how the viability of the cells are dispersed throughout these four strains. This is the table for the like result. The increase of NaCl concentration, determination, and a progressive and constant decrease of efficacy of the enzymatic activity throughout the incubation period, and in which we can see that however the purified TDC maintained a higher effectiveness even at a 15 percentage of sodium chloride concentration. The different means about 62 percentage of tyramine were produced in the absence of salt. So, and apart from that, different pH and at different NaCl concentration, the how much tyramine produced, we can see that in this table. uh, these are the this is one of the graph for which it is means mainly correlated with the tyramine production and the TDC activity about the different pH in the four different strains. So means there are the few means of the graphs are there. This is the result after the 2 hours, means how the tyramine is produced in four different strains after the 2 hours of the pH of the different four strains we obtained. And we have other means result after the 24 hours and all that, but just for the glimpse, I just means noted this result after the 2 hours. So we can see that all the means tyramine production rates for the four different strains were totally different, and their TDC activity were also be different as per the different strains and different hours. So all we can say that in conclusion of this entire experiment, the tyramine production by the cells is resuspended in the buffer system, highlighted the heterogeneity of the TDC activity within the enterococci. And after the research, we are allowed to correlate the specific mutation in the means tyramine production and its genetic sequence to their different tyraminogenic activity for the four different strands, and the role of the decarboxylase enzyme with the different substrates and the genetic organization was noticed for the first times after taking these experiments. And in future we will try to find out the phenolytic behavior of the TDC, by which we can also try to find out its correlation with the cell metabolism and the different environmental factors, and by these proper and in-depth understanding of genetic determinants and mechanism involved in tyramine production under the different chemical and physical condition will be observed in future in this research area. So we can know more about in-depth knowledge about how the tyramine production is occurs in different food while they are in the means aging and like a pickling process. So we can know the better understanding of this tyramine production. So thank you for means this research topic, I hope you all understand well. Thank you.